And now let me formally introduce our speaker. Dr. Steffen Tiedt studied medicine at Ludwig Maximilian University of Munich, as well as Harvard University. He's a leading expert in stroke research and has spearheaded some of the largest population health studies worldwide with early and serial blood sampling in acute stroke patients. His work focuses on identifying circulating markers that provide insights into the effects of stroke. And his lab utilizes profiling, ultrasensitive single molecule, and point-of-care technologies to identify meaningful signatures to improve stroke care and explore underlying molecular and pathophysiological mechanisms in experimental settings. So without further ado, Steffen, I'll pass the floor to you for your presentation.
Perfect. Thank you so much. I'm very grateful for the opportunity by Metabolon to present our research of the last couple of years. My name is Steffen Tiedt. I'm a senior physician at the LMU, and we are focusing, our work focuses on circulating biomarkers to help patients with stroke to improve their care by various means. The first talk focuses on how circulating metabolites could improve the diagnosis of stroke. The second talk will rather focus on identifying novel pathophysiological mechanisms of ischemic stroke. Why we are interested in that is because we see stroke on a daily basis. We see how stroke changes the lives of patients in an instant. Patients that were healthy in the morning develop from 1 second to the other loss of speech, or even sometimes worse, the comprehension of speech. Just imagine that for a second, that you would lose that ability to comprehend speech from your relatives, from your friends, for the next days, or potentially for the next months, or potentially forever. So we are very much inclined to develop novel tools to help these patients in a better way. Most of our research thus focuses on rather getting a better idea of the heterogeneity of stroke. We know that this is a large challenge for the diagnosis and treatment. And I give a couple of examples to this. We don't know the cause of stroke in more than 40% of the patients. And that doesn't only speak about that we don't know that our current classification systems are not ideal for identifying the potential atrial fibrillation in these patients, but that these classification trees are fixating on comorbidities and basically neglecting what happened in these patients. And blood biomarkers are obviously a tool to address that. We know that neural injury is undetermined in the pre-hospital phase and serially in the acute phase. Neuroimaging is obviously a great tool to have time-stamped assessments of how the brain looks like. But the brain is, the only organ that we don't have a blood-based biomarker for. We know that in stroke, most of the systemic effects are often undetected. We know that they are there, but they are not detected. We often neglect them. And we know that there is excellent experimental research in having identified various mechanisms explaining some of this heterogeneity, such as reperfusion injury. But again, we have no blood-based biomarkers to detect patients at risk, to identify the mechanism itself if it occurs, and to monitor it, and thus not the ability to develop novel treatments for this. For all these mechanisms, they share that we have no good diagnostic tools for them. But we obviously know that they're all kind of regulated, again, by our very genes, proteins, and most importantly, metabolites, which are at the very end of this grading scheme and the more dynamic molecules, which is important for an acute and dynamic disease such as stroke. More recently, also, we've learned that metabolites are not only these end products that are excreted, but are really important in governing multiple functional processes, and not to speak, of glucose, one of the metabolites that is known to everybody. More importantly, of course, also this is then again changed by the exposome. Interestingly, the proteome and the metabolome, of course, they represent the majority of our circulating molecules, and this now graph should give you an impression of how neglective we are, of what kind of information is right in front of our eyes if we make use of it. This is basically in comparison of the 20 proteins and the one metabolite being glucose that we assess on a daily basis, but there's much more that is circulating. We actually don't know the real number of proteins that is circulating, but it's could be 3,500, could be more. 25,000 are currently identified as part of the HMDB database as being circulating in the blood, and various research studies have made use of profiling studies, proteome, metabolome, to profile these, to identify novel biomarkers and to inform on local and novel systemic mechanisms. So we are inclined to make also similarly use of this for stroke. There's very few research on this so far, and to identify novel molecular signatures to increase diagnostic precision on this. In this part of, so in the first talk, I'm going to focus on real diagnostic, on something that has bothered me so much all the time when I see stroke patients in the emergency department. So there's patients coming in with obviously acutely developing symptoms when, let's say, the last 6 hours, but it's very hard clinically to differentiate between patients that have a real ischemic stroke and sometimes patients that have a stroke mimic. These are diseases such as patients with a migraine. These not always have a headache, but sometimes only present with their aura. Patients with an epileptic seizure, which are sometimes very hard to differentiate from patients with ischemic stroke, and other disease entities. Misclassification of these two entities might result in inappropriate interventions in either group, because you want to treat both accordingly, right? And withholding treatments with proven efficacy, such as if you are thinking that a stroke mimic, if an ischemic stroke patient is actually a patient with a stroke mimic, such as a migraine with aura, you're not going to treat this patient with TPA, which might improve the patient's outcome if it is really an ischemic stroke. And of course, inefficient allocation of resources, because patients with ischemic stroke are at much higher risk of developing future complications, and you want to protect them also from getting secondary strokes. We know what we do usually in the emergency department is that routine multimodal CT, being a non-contrast CT, being CT angiography, being also a CT perfusion scan, but then these have a limited value to differentiate the broad population of ischemic stroke patients from patients with stroke mimics in the first hours after symptom onset, when you need to make acute decisions for treatment. And this is basically here what we aim for. We want to differentiate this larger group of patients, really ideally to stroke mimics and patients with ischemic stroke, and patients with transient ischemic attacks, where symptoms then again wane over time within the first 24 hours. We thought by combining now the last two slides, basically, that the circulating metabolome might hide information on what happened in patients with ischemic stroke, and wanted to identify a signature, a metabolic signature for the pathophysiological mechanisms that are specific for ischemic stroke, and that might help us to diagnostically differentiate between patients with ischemic stroke and patients with stroke mimics. So for that reason, we started with comparing in a discovery and validation approach patients with an ischemic stroke here in the upper line and in red, and when they come into the hospital in the emergency department with a median of 5 hours after symptom onset. So really in the time window when you would like to see something and need to make an acute treatment decision. We compared this in the first approach with neurologically normal let's say subjects controlled. We identified using an untargeted approach, metabolomic approach, almost 700 metabolites. Our recent analysis, which I'm not going to present here, we know that this is now covering up to a bit more than 1,000 metabolites, but here we were with almost 700 metabolites that cover different classes, which you might all be aware of: amino acids, carbohydrates. These might be energy metabolites. There's a huge class of obviously different lipid metabolites. There's also various unknowns, and obviously also kind of external metabolites here. These can be differentiated into different pathways that these metabolites are belonging to, which you can see here. And there's an example here for amino acids such as glutathione metabolism, which is kind of very relevant for stroke because it's a redox pathway, lysine metabolism, tryptophan metabolism, which I'll come to in the second talk. We first, of course, looked for metabolites that are kind of detected in a relevant number of samples. And as you can see here, really the internal metabolites, not the xenobiotics, and also many of the unknowns, they were missing this step. But most of the metabolites from the body itself kind of made this step. We compared then basically these two different groups using a multivariable regression model, adjusting for potentially different baseline characteristics such as age, sex, vascular risk factors, and so on, and identified a bit more than 40 metabolites that you can see here really spread across different pathways and also classes. We then used our validation approach, being almost the same, a bit smaller set here, and found that almost all metabolites here were kind of validated. To differentiate now these two entities, which in a first approach, we tried to use different regression models, different machine learning techniques here, linear regression. And what you can see is that they're almost identical except for linear regression here, but led really by random forest, having an AUC value higher than 0.9. And this is, you can see here, the AUC and the ROC curve for the discovery and the validation cohort. Of course, we then wondered how the expression of these individual metabolites that are part of this a bit more than 30 metabolite signature, how this looks like, and whether they are correlating, because that might indicate that some of these metabolites are kind of redundant within the set. And this is also what we saw here in the seed map, where you can really see that most of the lipids here are kind of showing the same kind of profile among all patients, ischemic stroke and neurologically normal patients. So it indicated to us that there's a potential to eliminate some of them and to make it something that is clinically potentially more translatable, because obviously a signature of 3 or 4 metabolites is something that is much more tangible from a clinician's perspective and also from a laboratory chemistry perspective. But you might have asked yourself, why is he telling me about neurologically normal patients? He wanted to compare this with stroke mimics, and you're perfectly right. And this was our next step. So we added 2 different additional cohorts, derivation cohort where we compared this set of ischemic stroke patients with stroke mimics, and then a second validation step for the comparison of ischemic stroke with stroke mimics. And here we also used, you see, a similar size. This was propensity score matched cohort where we try to take care of also the fact that most of the stroke mimics are younger patients, and there's a bit of a higher proportion of female patients, a bit lesser proportion of vascular risk factors, and so on. But there was a cohort comparison that was matched for all these factors. Within this derivation cohort, we try to identify those metabolites which are most informative for making this separation between these two entities, the stroke mimics and the ischemic stroke patients. And as you can see here, there were 4 metabolites that kind of made that led up the ranking, which were ADMA, SDMA, pregnenolone sulfate, and adenosine. ADMA and SDMA are asymmetric and symmetric dimethylarginine. They are kind of 2, if you want to say so, isoforms of dimethylarginine. And we also followed these up with a targeted approach because we found that these have kind of different functions and that might be very relevant. And what we can see here is that really we see that both are very important and add on top of each other. By only focusing now on these 4 metabolites compared to the previous 30 or 40 metabolite signature, we still got between ischemic stroke and stroke mimics now in the derivation cohort, which was small, concise, an AUC value of 0.9, and that was higher in that cohort compared to the CT imaging that we usually do and serves as a gold standard in the emergency department. But that was done as part of diagnostic routine. That was not part of a prospective clinical trial, let's say. In the validation cohort here, in the second validation cohort, which was much larger, we still found kind of similar patterns. The metabolic set gave us an AUC of 0.91. The CT, based on non-contrast CT angiography, so identifying vessel occlusions, and CT perfusion, identifying potentially hypoperfusion, and any indication of that would rather be an ischemic stroke or a stroke mimic, we took as a true finding here, got only as a AUC of 0.73. We again were also then asked by reviewers to replicate this in yet another larger cohort of 500 patients overall, comprising 60-70% of ischemic stroke patients and 211 stroke mimic patients, which we recruited consecutively in our emergency department. Again, same findings, a bit higher value for CT, but again outperformed by the metabolite set. And I want to also mention here that this was not a linear regression model, but a random forest model that we developed here. We also wanted to see how this performs in patients that receive, for example, RTPA, and some stroke mimic patients also received RTPA, and we wanted to exclude those because that might be patients which were MRI negative, but sometimes there's the question of whether these patients were ischemic stroke patients, but you didn't really see a stroke then on imaging because you performed thrombolysis so fast. We excluded those, but that didn't really change the curves. And to go back to our original approach, comparing these with neurologically normal controls, we found that this set also still has a very high AUC in differentiating these two, which of course is no clinical question. Nobody needs a new diagnostic biomarker for differentiating patients with ischemic stroke and neurologically normal controls because you can just differentiate them by looking at them, of course. We wanted to see now whether these metabolites are indicating just patients at risk for stroke because they have a set of vascular risk factors, or because this is something as part of the acute phase of stroke. Because we didn't have any blood levels before stroke, nobody usually has, we compared this over time. So we looked at, for example, here now from neurologically normal controls, day 1, day 90. And what we found is that for all these metabolites here, for the 4 metabolites that basically comprise our set, we found a change in the acute phase and then return to normal levels at day 90, indicating that this is not something that is indicating a population at risk, these metabolites, but really associated with the acute phase. Upregulated or increased for these 3 metabolites and decreased for adenosine. We also looked at whether this is something that is anyway dependent on stroke etiology, which was largely not, except for SDMA, which has been shown before that they show higher levels in cardioembolic strokes, but otherwise these metabolites are really would be applicable across stroke etiologies. We looked at the relation with infarct volume, with final infarct volume, to further get any insights into whether these metabolites are related to the brain injury itself, or rather the vessel occlusion, the thrombus formation, platelet aggregation, and so on. And we found that only for pregnenolone sulfate there was a positive correlation, which we identify or interpreted rather as something that is, because pregnenolone is related to the cortisol pathway, is something that is related to the stress response, and patients with larger strokes show a higher stress response compared to its directly from the brain itself, which we cannot say right now, but at least most of the other metabolites were not showing this relation. We also looked at the relation with pre-processing analytics, the time from sampling to freezing, and there was no relation for these 3 metabolites, but one for adenosine, which has been known before, which is a very short half-life, and you see already here also this decrease. So if that enters clinical routine at some point, you need to be fast in getting the sample to the lab. We looked at how our set outperforms other studies in this field and identified in a systematic review here from more than 3,000 records, 5 studies, which kind of took up a similar question. They mostly or almost exclusively compared ischemic stroke patients with neurologically normal controls. These are the metabolites from these studies, and we had looked into these metabolites in our data and identified that for the comparison to stroke mimics, because these data sets could not give us anything on stroke mimics, and found that all these AUC values were lower than the ones that we found for our 4 metabolites. That led to this paper, which has been published in the Annals of Neurology and also received excellent paper in New Science Award by the RNet Neuron Consortium. And with this, I finishing up this talk already. You see up there also for the second talk my contact details. I'm happy to take any queries which cannot be answered in the questions. On this part here, there were many. We have a group that is comprised of outstanding young researchers, and I'm grateful for every day that I can work with them. People that have left us in the past, but have contributed considerably to this study, and also the one that I show in the second part, in the second talk, our collaborators on site here really at LMU and nationally and internationally. Thank you for your attention.
Thank you very much, Steffen, for this first part of your presentation. Fascinating work. Very interesting to see how these biomarkers perform in the clinical setting. A couple of very quick questions. Have you looked into other omics to complement your metabolomics data?
Yeah, so we have currently expanded our cohort or set up a new cohort, which we call the PROMISE, the Precision Medicine and Stroke cohort, where we not only have started with 70 versus 40, but we follow 500 patients from day 1 to day 7, and where we also have got these metabolomic data, but are also currently performing and expecting the proteomic data to be finished this year, and are very much looking forward to the analysis of not only individual proteins, but really also the integrated analysis.
Perfect. Yeah, it will be very interesting to see how these biomarkers behave in combination with regards to discriminating between the different patient groups. And another question, what will be the most appropriate next steps into developing these biomarkers into a point of care test for clinical applications?
Yeah, I'm still looking forward for also validation studies that are not coming from us. We had several inquiries also to help setting up other studies, but in the end, there's the need also to have similar data on the same metabolites from other cohorts, let's say, and if that is successful, we would of course try to aim at develop a point of care test, which we have already reached out to our on-site lab department, but which is tricky, as you know, sometimes for metabolites. Depends a bit on their class and about their chemistry, the chemistry behind, but we're on it.
Excellent. Thank you so very much. And I will take the next few minutes to give you a very quick overview of our technology and our solutions. Okay, so again, thank you very much for joining us today. I'm just going to spend the next few minutes to give you an overview of what we do at Metabolon and how our solutions can be used and can be readily applied to enhance metabolomics research. Now, Steffen has given us a fascinating example demonstrating how metabolites can prove to be of great utility in the clinical context and how findings like this have the potential to be integrated into clinical applications and complement existing tools in order to improve decision-making at critical moments, such as those in the emergency department. Now, metabolomics can, of course, provide insights beyond just prediction of diseases, as it offers a broader spectrum of advantages. Metabolomics as a technology enables us to get a better understanding of disease biology, facilitate patient stratification, as we've seen by Steffen's presentation, and also direct the development of new therapeutics. Now, tackling these challenges and moving the needle in the translational space with metabolomics requires access to high-quality data and insights. And Metabolon offers effective solutions across 2 distinct categories of products: the Metabolon Discover family, as well as the targeted panels for metabolite quantification. These panels have been designed to answer different types of questions, as well as address different research or application needs. Our global discovery panel, our approach to untargeted metabolomics, offers unrivaled metabolite coverage, analyzing as many metabolites as possible in a biological sample without prior knowledge of what specific biochemicals may be present. So with this discovery approach, we can capture changes that are related to a variety of factors, such as our genes, the environment, our diet, the microbiome, really delivering the best possible representation of the phenotype. We've also recently launched our focused discovery panels tailored for specific areas of research, such as biological stress, liver and kidney diseases. And these panels analyze subsets of relevant metabolites, and it also come with a suite of advanced data visualization tools that we have developed and are designed to significantly enhance the interactivity and the interpretability for investigators. And then we have our targeted panels, which basically build upon discovery work, providing more precise and accurate measurements of a select few metabolites of interest. We currently offer 22 targeted panels and single analyte assays, including our lipidomics panel and our tailored diabetes and inflammation portfolios, offering absolute quantification of important biomarkers and specific metabolic pathways. Now, Metabolon truly sets the standard in metabolomics analysis, excelling in coverage, identification, as well as data quality. The main driver that enables us to offer the quality of services that we do is our extensive reference library of authentic chemical standards, which we have either purchased or synthesized ourselves. We've run those authentic standards on our platform, and we have information on their exact fingerprint. Our library covers over 5,400 metabolites, and it allows us to offer the highest level of annotation confidence as well as depth of coverage. Now, capturing metabolites that come from over 70 major metabolic pathways with our discovery approach, we can really deliver unique insights on the many and diverse processes that occur in a biological system. Next, I'd like to quickly go over our pipeline of work and how working with Metabolon looks like. So we offer personalized guidance at every step to just ensure the best possible result for our investigators. We like to work with you early in the study planning phase to ensure appropriate study design, which is by far the most critical step in metabolomics, determining outcomes as well as the success of the study. With that said, when the samples come into our labs, we'll perform the extraction and have the samples ready for analysis. Now, we know that biological matrices are extremely diverse, rich, and very complex in their biochemical makeup, the biochemical content, as well as the physical chemical properties of these analytes. So detecting these molecules in itself presents us with a great challenge. And the only way to really capture this biochemical complexity is to use different and complementary data acquisition methods. After the samples have been prepared and extracted, are divided into 4 aliquotes and are run over 4 different analytical methods to maximize metabolite detection and coverage. The data that is generated, which is pretty rich in noise, then undergo thorough processing as well as quality control to eliminate part of that noise and perform library matching and metabolite ID. The final step of our pipeline is bioinformatics and data interpretation, where identified metabolites are then mapped into biochemical pathways so that we can get a better understanding of what is happening in a biological system and start making recommendations for follow-up actions. As I mentioned, mass spec data is very rich in noise and artifacts and redundant signals, which might not always be of important biological origin, so they may not have anything meaningful to contribute to the study. So I'd like to take a minute and go over our chemocentric approach, the approach that we use towards extracting biologically meaningful data and significant metabolites from these very noisy data sets. Each experiment that we run generates about 150,000 ion features. But having 150,000 ion features or peaks does not necessarily mean that we have 150,000 metabolites in our samples. So what we do is take those 150,000 ion features and search them through our library to identify the source of these features and match them to an ID. For each of the compounds that exist in our library, we have information on their retention time, their mass, and their fragmentation pattern. And this allows us to filter through all the noise and extract appropriately annotated metabolites. And this gives us the benefit of reducing false discoveries, which is a major issue in these types of experiments that generate these large volumes of data and allows to only use relevant metabolites to further examine and take into statistical analysis and downstream biological interrogation. After following this data reduction step, where we've removed the noise and artifacts from our data, we can deliver confirmed metabolite IDs in a format that is readily available for statistical analysis, saving investigators significant amounts of time and resources, which can dedicated or invested to downstream biological interrogation and inform further research activities through these actionable insights. And after we've completed the process of metabolite ID, the last part of the pipeline is, of course, the interpretation of the data. So our services do not stop at data generation, but we can help with taking the results beyond just a data table of metabolites that go up and down. So we're not just providing you the pieces of a very complicated puzzle and let investigators figure out what to do on their own. So with the help of our discovery and translational sciences team, our biochemistry and metabolism experts, and the wealth of institutional knowledge that we've built over the years, we can help interpret these complex signals that come from these very rich data sets, identify and communicate key findings of a study, and provide guidance towards the next steps and actionable insights based on what the data is telling us. So that's all I had to share with you today regarding our technology and our solutions. Please feel free to use the Q&A box to type any questions you may have. And with that, I'd like to pass the baton on to Steffen for the second part of his presentation, where he will discuss some of his work on identifying metabolic signatures of local and the systemic effects of stroke.
Perfect. Thank you so much. Yeah, as I said and finished up the last talk, there's much more than the pure diagnosis with stroke. And I'm going to shortly repeat this again here. It's about the etiology of stroke. It's about to follow brain injury after stroke with a blood-based biomarker, and that would be obviously so informative for other brain diseases where it's much harder to identify the injury itself, such as people with brain trauma, such as patients with intracerebral hemorrhage, where it's really hard to say how many neurons died behind this blood clot, patients with subarachnoid hemorrhage. We speak about the systemic effect and about also existing preclinical concepts. So it's really overall it's about local, so the brain and systemic effects after stroke. And we've recently also wrote a review about this. If you would like to look at it, I'm going to again start with this slide because I think it clearly shows the difference of what happens in the acute phase here at day 1, and we also sampled these patients at the next morning, so right at 24 hours. And to my very surprise, what we found is much more that is going on between intra-individually between day 1 and day 2, arguing for also that stroke is such a dynamic disease setting where it's so important to really know when to sample, also if you look for a biomarker, and not to say, I do this in the first 24 hours and it's all kind of the same. That's not as we see here. Much more was different between day 1 and day 2 compared to healthy controls without any stress response in an outpatient clinic setting in patients in the emergency department. We look now at this at various levels. You can look at this obviously, and you might all know about it, but I'm going to show you our data on stroke here on a global level and look at the variability of metabolites. And we looked here at Z-scores of how they are basically developing over time and the variability of between healthy controls, day 1, day 2, and day 90. And what you beautifully can see here is that there's stroke itself induces a global variability of metabolites, as you can see here behind the Z-score of 2 or -2, much more individual data points where behind left or right of these lines compared to healthy controls, continuing here at day 2, similar, and going back to control levels kind of at day 90, again arguing that the dynamic of the acute phase are highly important. And we also found this similarly you're doing a PCA, a principal component analysis, which looks at the data without knowing anything about the groups and then assigning colors to the individual data points only after the analysis. So the light gray is the healthy controls. You see that how the shifts away, there's a metabolic change at day 1, it further shifts away at day 2, and then returns to kind of control group levels at day 90 again. We looked at individual classes, amino acids here, lipids here, and we found that this is a bit of I kind of had to take it apart for this presentation. So this heat map is 3 lines, and you see individual metabolite names here on the left. You see day 1 compared to healthy control changes here. You see day 2 compared to healthy control changes here, day 90, and then the class of the respective metabolites. So grouped by class, and here then ordered by how they are different at day 1 compared to healthy controls. And then this metabolite here is exactly this metabolite here is exactly this metabolite here. And you can follow individual metabolite developments from day 1 to day 2 to day 90 in relation to healthy controls. For serotonin, you can say, for example, that it's increased at day 1, further increases at day 2, and returns kind of to control levels here at day 90, because this white space indicates basically a beta value from the linear regression model of 0. Other metabolites, which I'll come to in a second, are indole propionate, which was decreased at day 1, further decreases at day 2, and then kind of normalizes, kind of still downregulated at day 90. You see also cortisol here, which was highest among all metabolites in its upregulation after stroke at day 1, keeps that level at day 2, and then returns or even is lower at day 90 compared to healthy controls. That is nothing new information that I tell you here. We know that the stroke elicits a stress response. Patients show higher cortisol levels. That is related to outcome, that is related to infarct volume, and so on. I want to mention a couple of other metabolites here, such as beta-hydroxybutyrate, which is one of the ketone bodies, which we found elevated here, further elevated at day 2, so 24 hours after stroke, and then returned to control levels at day 90. And you will see that metabolites again in a few slides. We then looked next at pathways, so global class pathways. So there is a smaller set of metabolites assigned to one pathway. And we used MetaboAnalyst, which is both available as an R package, but also as an online available platform where you can upload your data. And we use that tool to map these pathways and to investigate changes of metabolic pathways between ischemic stroke and healthy controls at day 1 and day 2. Changes of the same pathway are indicated in light red at day 1, dark red at day 2, and you see here a Y and an X-axis. The Y-axis indicates the minus log p-value, so the higher, the smaller the p-value, and the pathway impact indicates that, of course, we are for no pathway or for very few pathways able to track all metabolites. But for most pathways, we are really having, let's say, 3 out of 6. And then there's a central metabolite and a few peripheral metabolites. And how central these metabolites are that we are assessing in our data set to the overall pathway integrity, this is indicated by the pathway impact. So a pathway impact of 1 shows that you have all metabolites or even the most central metabolites. So you basically can make a large, deriving large kind of amount of interpretation of this pathway, while pathways which a pathway impact of 0, you might have 1 out of, let's say, 20 metabolites, and you only have 1 peripheral metabolite. So usually one skips those which are here at this range kind of and focus on this one. But overall, we investigated a bit more than 60 metabolic pathways where our untarget metabolomics set kind of covered, and more than 75% of these pathways were significantly different, indicated here by this dashed line after stroke. And you see this is here led, I have put a few numbers here, such as the fatty acid biosynthesis, steroid hormone biosynthesis, which is probably covered mostly by cortisol, the levels that I showed you on the previous slide. But we also see synthesis and degradation of ketone bodies. I also mentioned this on the previous slide. And we see also a large amount of pathways that are related to aromatic amino acid pathways. And I'll come to this in a second. This is why I highlighted it here. And for most of these, we see that there's a, if you want to say so, I know statistically that's possibly very wrong what I say, but there's this decrease in p-values here, or an increase of the minus log p from day 1 to day 2. So it shows the dynamics, of course, so the light red and dark red do not overlap so much for these metabolites, but there's a development of these pathways. We next wondered, because we compared this to healthy controls, what actually is stroke specific. And of course, what we thought about is that stroke specific is the brain injury itself. So what we wondered is whether there's any relation of the pathway changes to what we see in the brain, so the extent of the brain injury. And for this, we focus on the change of day 1 to day 2, because we know that patients have very large inter-individual differences, and focusing on the intra-individual difference between day 1 and day 2 takes away a large amount of this variability. Statistically, it makes it easier to identify relations. And this is what we found here. There was 8 pathways that were related to infarct volume, so that might indicate that either these are biomarkers for brain injury, or they have potentially also something that is influencing the extent of brain injury. And these were really focusing on amino acid pathways. And again, here you see also a few aromatic amino acid pathways, tryptophan, phenylalanine. Again, that's coming up also on another slide, but also redox pathways such as nicotinamide metabolism, which is, as you might know, one of the key signaling molecules, not only for redox metabolism, but for many, many different other pathways, NAD. We looked at, because metabolic analysis gives us also the opportunity to map these pathways to organs here, tissues, cell types, and cellular compartments. And of course, you might say, how can you say with a prospective observational cohort that this metabolite now derived from the CNS? Then you also shows brain. That is even not exclusive. You're perfectly right. And this is obviously a mapping approach, a bioinformatic approach, and a non-exclusive approach, basically leveraging existing data sets from cell culture, from in vivo tissue analysis, but cross-sectionally, it gives us an impression not on an individual metabolite level, but rather, what of the metabolic change that we see is, is deriving from CNS or brain or other tissues, and it makes it possible to derive pathophysiological information from it. And importantly here, or intriguingly for us, we found that on an organ, tissue, and cell type level, those tissue notations or organ notations, cell type notations that are associated with the CNS or the brain itself, because stroke affects the brain, these always ranked highest compared to others. But as you know, stroke also has systemic consequences, so we also found others. And this is so important for us as researchers investigating blood-based biomarkers, because in the first 24 hours, we see here apparently a metabolic response that is derived from the CNS or relates to the CNS, compared to, for example, neurofilament light chain that we investigated some time ago, where we see that this protein that is so specific for the brain and gives us face validity that it derives from the brain if we detect it in the serum, that this only changes after 50 hours after stroke onset. So metabolites, we know from this analysis, it indicates that metabolites smaller, passing the blood-brain barrier potentially earlier compared to larger hydrophobic proteins, which are, on a secondary, basically, potentially even only leaving the brain. We tried to take all these individual metabolites that you looked in the circus plot on the previous slides, to take into a network and to derive something that could tell us about how these metabolites relate to each other, to identify potentially mechanisms behind the overall signature beyond the pathway analysis. And here you can see every individual dot represents one metabolite. The ones in red are higher after stroke, the ones in blue are lower after stroke, and the lines are, of course, correlations. Gray lines are positive correlations, blue lines are negative correlations. So you see there's many, many positive correlations. And you see clusters of metabolites. What are these clusters? These are, of course, metabolites that belong to the same class or kind of same pathways. That makes sense because metabolites from similar pathways or classes undergo similar enzymatic degradation. So one enzyme is responsible for processing various metabolites at the same time. So they kind of have similar expression values. Here you see this beautifully for long-chain fatty acids, but you also see this for various other classes here, how they cluster very nicely together. But we also found something, and we haven't looked at this even in detail, and this is only one preliminary data set of a cohort of this overall 500 patient cohort that I spoke as a reply to Natasha's previous question. We looked at this. We now first found that these two key nodes here, these are kynurenine and tryptophan. And I mentioned to you several times tryptophan, and you might wonder why did he do that. So tryptophan and kynurenine, they seem to be relevant in regulating metabolic changes and connecting metabolic changes, potentially also having a coordinary role. We looked at this because we know tryptophan is an essential amino acid. So there's limited amount, and there's basically, you can, I would say, not 100% safety, but you can say that almost exclusively some of these metabolites that are derived from tryptophan are coming from the host, from our host cells, and some are coming from microbiota. So mostly the indoles, they derive almost exclusively from microbiota, and are not having anything to do with the host. And you can easily see here this difference that basically the host process metabolites, they did not basically change at all after stroke, while microbiota-related metabolites, they considerably decreased after stroke, indicating that there's something that is specific for microbiota in terms of metabolic changes that we see after stroke, indicating then again, obviously awaiting several future studies, but that we could look at metabolites to follow gut dysbiosis after stroke, and that would give us on a clinical care basis so much more insights, because we know that stroke bilaterally interacts with the gut microbiome in a way that stroke changes the gut microbiome, but also the gut microbiome functionally impacts by immune cells and metabolites on the stroke outcome. And there's some beautiful work of Arthur Lees and Corinne Benakis out there that is published well. So that could give us an opportunity to look just as a blood sample to know about gut dysbiosis. And that is, of course, also relevant because our patients receive so many antibiotics, which might again change the gut dysbiosis and so on. We also looked at other metabolic pathways, mostly here aromatic amino acid pathways, which are also under control of the microbiome, but where it's not that exclusively clear which ones derive from the host, which ones derive from the microbiota. There's a good overlap, but we see a general downtrend here. And also for bile acid metabolism, where we know host does primary bile acid processing, secondary bile acid rather derived from microbiota, but of course there is feedback. But again, a general downtrend indicating that also these might integrate into the metabolic change that we see for microbiota. Going back to this network, which we found fascinating to look into, and we found that there's something that I personally couldn't make sense of. I, of course, study medicine and not biochemistry, and I'm an expert in biochemistry. But what we found is that long-chain fatty acids here, this large cluster, there's apparently a negative correlation with these glucogenic amino acids, proline and alanine, and this ketone body beta-hydroxybutyrate here, which clusters here. I personally need to go back to the textbooks of biochemistry, and what we found is that you might all know that there's these relate kind of in terms of energy metabolism with each other. We know that long-chain fatty acids are undergoing beta oxidation to give rise to acetyl-CoA, which enters the TCA cycle to give us energy. While there's also obviously amino acids which can provide oxaloacetate, which can be used for gluconeogenesis and so on to generate glucose. Stroke is obviously a disease where the brain is affected and where the energy metabolite glucose is highly relevant for the brain and to be delivered to the brain. So we know energy metabolism is important for stroke. What do we see after stroke in our metabolomic profile? What we found is that here in the acute phase, 5 hours at the very first time point, day 1, we found this change here with elevated long-chain fatty acids, elevated metabolites of the TCA cycle. Of course, this is only indicative because we look at the blood sample that, is integrating information from various organs. But overall, the signature might tell us that we have an acute catabolic response. And 24 hours, this picture completely changes with stagnation of long-chain fatty acids, increase of beta-hydroxybutyrate, which you also saw on the heat map, a decrease of TCA cycle metabolites, and an increase of proteolysis. This is what this signature indicates, indicating this potentially a delayed catabolic response and proteolysis and ketone body synthesis, which is highly relevant for our patients because we know that these stroke patients, that 20% of them lose weight upon discharge in up to 1 year. It's not as extensive, obviously, as in brain and burn injury, but we know that they have catabolism. Some of them are remain fasting for quite some time because of dysphagia, because of other also upcoming surgeries. And usually we don't care because, or the traditional knowledge says everybody is able to fast for a few days. But in such a setting where we have a brain disease and an ongoing infarction, which is not set within the first 2 hours, but which is ongoing and growing within the first 24, 48 hours, where the penumbra is lost, where the brain needs energy, it might not be the best idea to keep patients fasting. And these metabolites here, and we're going to look in this in detail, and need to develop this much more, might give us hints to identify a biomarker set to identify those patients at risk, where this is particularly important, to identify the catabolism itself, to monitor the catabolism, and to potentially also prognosticate outcome. This is just an outlook, and I mentioned on this in the previous reply. We're going to expand this much more with our precision medicine and stroke study, which was a prospective and registered observational study where we included patients with a rapidly developing clinical signs suggestive of stroke within the first 24 hours. I believe it's the largest study on this matter worldwide because we recruited 502 patients and did serial sampling day 1, day 2, day 3, day 7, and on a sub portion also of day 90. And we combine this serial molecular profiling, metabolomics, and also proteomics, as I said, with a longitudinal imaging approach. We collect imaging data from the acute phase when the patients come into the hospital, giving us an insight into how much brain tissue is already lost, how this develops over time to a final infarct volume, whether these patients sometimes also show secondary ischemic or hemorrhagic events, and so on. This is one of our main goals, to identify something that helps us to track brain injury, and this is already very beautiful initial work from Naomi Flegels, who works on this data set, where we see that our data set here covers really most of the brain. This is a lesion prevalence map. Any color except for this light gray indicates that we have patients with an infarct in that region in our cohort, with obviously here predominance in the right territorium of the middle cerebral artery, a bit more than the left side, but not a clear bias that we usually see in larger studies. Also, this study will allow us to track individual brain regions to metabolic changes in the body. We're going to look for these things and also try to identify surrogate markers for treatment effects, and obviously look for the association with clinical outcome. And overall, our idea really, and this is taken as a suggestion and to you, and would look forward to discuss this with all of you who might also have experience with other disease entities, but it should take into everything together from the first talk and the second talk that we want to look at, and the heterogeneity of stroke, be able to develop biomarkers to stratify it in more defined way using genomic, proteomic, metabolomic data, but also a deep clinical phenotyping approach to develop different molecular signatures for the stuff that I said on the first slide in the two talks. Because I really think that this is needed. Everybody focuses so much on treatment, and treatment obviously is the most important thing that we need to improve for patients. But in most instances, I personally feel that we just lack the biomarkers to recruit the right patients, to identify the right patients. And for stroke, this is so true because there's this large translational roadblock between experimentally validated or verified treatments which worked in mice and have never worked in humans. There's a paper in Stroke that says 1001 experimental treatments and 0, or maybe they counted other places into it, and 1 that worked in humans. So we need to do much better on bridging this gap, on translating experimental stroke research to clinical stroke research. And I personally believe that we need much more defined and fine-tuned biomarkers to do that. With that, I'm finishing up with the same slide, but I don't want to say the same words again. So I'm thanking you for your attention and for keeping with us. Thank you.
All right. Thank you so much, Steffen. One question is whether you've looked into diet or other comorbidities, especially diet, to look at the effects of those lipids that you detected.
Yeah, so what we are trying to do now is to, in our cohort at least, to also, in PROMISE, we have done that to track the intake, the nutritional intake of these patients and to correlate it with the changes that we see. We ourselves haven't done any work in patients that were in healthy controls that were kept fasting for an amount of time. But there's beautiful work out there by colleagues that focus on this.
Mm-hmm. There's another question. Have you used the time from symptom onset to day 1 blood sampling as a co-founder besides sex and age in your models? If a day makes a difference, could the first hour or minutes have different profiles too?
Yeah, I think that is a beautiful question, and I think so. We have done that, because we see that there's obviously a dependence for any biomarker, or really most biomarkers, because we believe that stroke is an acute disease. Any mechanism in the acute phase changes, so it's so important to focus on to take into account this time after symptom onset. Because anything else that remains the same and is independent of the time of symptom onset might rather indicate a population at risk and is not related to the acute phase. So it's important to take that into account, because for most of these things we see changes, and this is what we've done also in these models.
I mean, considering these changes, have you maybe used some of these metabolites to maybe predict other outcomes such as recovery or survival?
Yeah, right. So in our initial data set with 70 patients and then the follow-up, we were not powered to do that, but in our PROMISE data set, we are powered to do that, and we place particular attention to that, to collecting these outcomes. Not only, most importantly, functional outcome using the modified ranking scale score, which is 90-day MRS score, is the key outcome variable for all clinical trials in the stroke area, but also at discharge or day 7. But also morphological outcome is in fact volume. And what we see is really that, I can maybe give an initial glimpse into that if we just look at simple associations of metabolites with outcome, what we see is that we see an upward trend of the cumulative number of metabolites from day 1, day 2, day 3, day 7, indicating that, giving us much, face validity of that, what our cohort works. It makes sense to look at metabolites, because of course stroke the closer you get to day 90, day 7 is closer to day 90 than day 1. At day 1 you have many dynamics still going on, so the further you go, the more associations you get. And this was beautiful for us to see, and it's actually quite a high number, so it's several hundreds of overall 1,100 metabolites.
Which I guess is going to make it very challenging if you want to, develop this into a point of care test where I guess metabolite quantification becomes an all-important matter there.
I think it's actually very exciting because it, you know, I personally believe that there's not much value in developing or identifying a metabolite that per se that is associated with outcome, because every clinician, every patient would have a hard time believing that, you know, telling him or her that we measured this metabolite and now your outcome in 90 days will be like this, and taking it as something that is not about to change because the biomarkers are good. I don't believe that this is happening, and this is also leading us somewhere. I personally believe that this is that there's a lot of pathophysiology behind, so there's mechanisms behind, and we're going to need to tease these apart. And then there's probably from the several hundreds, few that are, you know, potentially speaking for gut dysbiosis. Some are on catabolism, some are on totally undetected mechanisms. We hope that there are some for brain injury itself, and then this list will be narrowed down. Many will correlate with each other, narrowing also down because usually, you know, if they correlate with each other, there's one with highest value compared to the others, and that's how I see it.
Right. Perfect. Steffen, thank you so very much for joining us today. It's been wonderful having you present our research. We're very happy to be playing a role in this very, very important work. So thank you very much for attending. And yes, we'll see you in a couple of weeks' time in our next webinar. Thank you very much.
Thank you, Natasha. Thank you all.